to note. For example, you might have a lesion that become hypoplastic, okay, which means more cell number which become dysplastic, okay, dysplastic, which is darker nucleus staining. The cells sort of like pile on top each other. They kind of lose contact inhibition. We call that. They become carcinoma in situ. I would say that with the carcinoma in situ, the stroma is present. So it's still considered as very benign, right? Benign. And then it becomes metastatic cancer. Metastatic cancer. All right. So this is typically, uh, my research on colorectal cancer, and this is what the events that we see. You can define it as carcinogenesis. They become hypoplastic cell, dysplastic cell, carcinoma in situ, and metastatic cancer. Now, within the hypoplastic cell here, right, there are many cells, but they're not all the same, by the way. Okay, in fact, a lot of these cells are different from each other. Okay, different from each other. They're not the same, and so given whatever the environmental pressure is put on there are some cells that then grow uh, that will grow selected to grow, for example. Okay, we have the selective advantage, right? And so then you can say, oh, it seems like one of the feature of cancer is that is very very similar to what Darwinism is that in Darwin, um, you know, theories of evolution is that there is, first of all, you have changes within the population, and the environmental pressure select for the species that is give it the advantage of surviving. So this is very similar. You have the diversity there first, right? This is the diversity, and the environmental pressure then is selecting out those that do not survive in this environment will not do, but those that can survive will proliferate. And here you can see that the yellow one proliferate or the orange ones proliferate. Okay? And so one of the feature of this malignancy is that there is heterogeneity. Heterogeneity means there's different population. If it's just one population, then it is known as homogeneity, but it's not. It is heterogeneity, and that is not only you have one, but you can have several that's selected to divide. And those cells that do divide, typically, in fact, you can look at the type of tumor, okay? Um, and cells that are divide usually we can see them during the process of mitosis. That might be at the middle of the metaphase, for example. Okay. And so this tumor has 3 cells that we know that it's dividing because it's in the metaphase. Will divide much faster than if a tumor that you see that, but you don't see very many mitosis. So depending on the number of mitotic cells, okay? So in this case, you see that there are 3 mitotic cells, right? Then that means that this one will have faster growth than this one that has no mitotic cell or cell that is dividing. Okay? So the growth rate of a neoplasm depends on the number of dividing cells. Another word for dividing cell is known as mitotic cell. So, right? So in the future, if I say, this tumor you can see a lot of cells in metaphase, then that means that you know that it's going to be rapidly, it's going to divide rapidly, as opposed to a tumor that you do not see any mitotic cells at all, then they're kind of like slow growing. All right. So when you're looking at the lesion, you can look at the cells and finding how close is it to the parenchymal, how close is it to the original cell. So, all right, what I mean by that is that, let's say you have a lesions in the liver, and you look at the cell and say, oh, does it look like hepatocyte, which is in the liver, or is it undifferentiated? Okay, lesser degree, if you of differentiation, if you will. So in other words, either greater differentiations or lesser differentiation. And I would say this: in the tumor, right, or a lesion, let's say this, differentiated cells in the tumor is easier to treat than undifferentiated cell. Okay. If they're undifferentiated, then they kind of like go back to their original state. But differentiated cells tend to be easier to treat than undifferentiated cell. Okay? I can tell you that. Here's an example. One of the problems of treating breast cancer now is that there are breast cancer cells, we call it triple negative. Okay, triple negative red blood cell, triple negative breast cancer cell. And triple mean because it has, they don't express estrogen receptor, progesterone receptor, or human epidermal HER, okay, or receptor. HER is stands for human epidermal growth factor. All right, so triple negative meaning that they do not express three things: estrogen receptor, progesterone receptor, or HER. Okay? HER stands for human epidermal growth factor. Certainly, cancer cell, breast cancer, require EGF. So typically we call this EGF, epidermal growth factor, for it to grow, right? And so somehow the cancer cell now doesn't need epidermal growth factor; it just grows. They could produce more of it, or they might constitutively active the receptor. That's another component of you can think of. In other words, let's say here's a tumor cells, and here is the EGF, epidermal growth factor receptor, right? And usually, if there is an epidermal growth factor binds to this, then you get an amplification signal, usually phosphorylation that is occurring like this, right? But somehow it's possible then that the tumor doesn't need epidermal growth factor. In other words, there is a mutation in the receptor, or somehow this receptor can activate, can undergo this pathway in the absence of epidermal growth factor. Okay? There's a mutation, so we call that constitutively active receptor. Constitutively active receptor. So in other words, the receptor is activated. It's always on. It doesn't need the epidermal growth factor to turn it on. It's always on. And so this cancer then, may have this, or sometimes the cancer do not have that. Right? The cancer do not have it, or do not have this, or do not have estrogen receptor or progesterone receptor. If they don't, then we can say, oh, they're not differentiated, because one of the characteristics of breast cancer, if they differentiated, they would produce estrogen receptor. If they do, then it's easy to treat because you can give it tamoxifen. Okay, tamoxifen is the estrogen, is the estrogen analog. Okay, so tamoxifen is an estrogen analog, and so if the cell produces estrogen, so we call it ER positive, right? You can give the patient tamoxifen and they're responding to it. They're like, okay, if the cancer cell require estrogen to survive, right? If it's require estrogen, it need estrogen receptor, then you treat it with tamoxifen. But if the cancer do not express this estrogen receptor, right? How do you give it a tamoxifen? Won't, you know, it doesn't express, right? And tamoxifen won't do anything to it. All right, take home message: differentiated cells are easier to treat than undifferentiated cells, right? So when you look at a lesion and say, oh, it's expressed this, it's this, boom, that's good, because it means it's treatable. If it doesn't, then it's not treatable. Neoplasm, certainly every cancer cells require nourishment. Malignancy, so associating malignancy means metastatic, um, that you have to upregulate the stromal. That's usually invasion metastasis, a hallmark of malignancy. And another important thing is that there are immune cells that seize these cells, right? They're baying, right? They're surveilling, uh, so they make sure that they keep the cell from uncontrollably. And typically, usually, if you have that problem of the suppressor T suppressor cells not working, or T regulatory is not working, then you get cancer, if you will. Okay? But in general, immunosurveillance is around to fight against that cancer. But if that goes away, then you get full blown. The couple of years ago, um, there was a Nobel Prize was awarded to um, um, what's his name? But he's working on I can't think of his name. It's Alistair. Yeah, Alistair, James Alistair, who works on T cells that keep these. The T cell has a ways to put a break so that it doesn't kill your cancer cell, for example, or doesn't kill your normal cell. And so what James Alistair did was that he tricked the system that's involving CTL receptor so that now it removed that break, and so the T cell, cytotoxic T cell, can go and kill the cancer cell. So this is the way of using your immune system to kill the cancer cell. In that case, it's a cytotoxic T cells. Okay. James Alistair work. Okay. Carcinoma. When you say carcinoma, particularly in situ, is still confined, right? It's considered benign. I would say then, so this is probably the stroma here. This is stroma here. And when these cells break away from that, because they're upregulating enzymes, then they become the metastatic cancer. This is kind of, it's gonna be there. It's pre-malignant right here, so that's going to break away and it's invade. Particular here, I think this is if this is cancer down here in the stroma, that's metastatic right there. Okay, that's metastatic. Um, and you see here's the basement membrane. So the basement membrane is a stroma. Okay, it's a stroma. All right. Normal epithelial, so basically similar carcinogenesis, dysplasia, carcinoma in situ, and you can see how the basement membrane is intact. It has been destroyed, right? Invading, right? Uh, and invading can either goes into the blood or can go into the lymphatic. Okay. Um, and whether they go into the lymphatic and in the blood, it depends on what kinds of tumor. In the breast cancer, usually they're invaded to the lymphatic more, if you will. But they can go into the blood, but they tend to go into the lymphatic first. And so when a female feel lumpectomy, feel a lump, and if it's suspicious, you can remove it called lumpectomy. But when you see a lesion, particularly if the skin is attached to it and it's very wrinkled, then that's not a very good sign. We call it stell-like, okay? And you can see this in MRI, for example. And to note whether it has been metastasized or not, what you have to do is you have to remove the lymph node and see whether there's cancer cells in the lymph node. Okay, um, and so that's part of the staging. So I'm going to talk to you to differentiate between grading, which is an anatomical exercise, versus staging, which is the clinical definition. And staging depends on T and N. T for tumor size, N for node, whether it's node positive, and M for metastatic site. So we call it TNM. Okay, TNM. All right, doubling time. So essentially, here is this is pre normal person and 20 years old, smoker, a pack of cigarettes every day. And you can see that normal cell over time, years, now have reaches, you only have one malignant cell that then divide, even though it looks like it comes from one, but it's changing constantly here. Okay, this is heterogeneity. It's not homogeneity. Um, it could be one from here, or it could be other cells that give rise to another population. But note that by the time you're getting a tumor size, it's multiple clonals, if you will, right? Or they're constantly changing here. So they're no longer homogeneity, but heterogeneity. And once it reaches the 1 centimeter size, okay, 1 centimeter size, usually it's the finger length. Yeah. And from that so from one cells to this one centimeter size, it could take years, 20 years, 30 years, 40 years, or 50 years. But once you have this one tumor size, 1 centimeter size, to reach this to metastatic site, it's in a few years or something. You see how the graph of this thing is like kind of like exponentially, if you will, right? So it could take years, but once you reaches these 1 centimeter site, and it just basically boom, right here, exponential phase growth. Um, and so from here, from 1 centimeter size to metastatic site, it just takes months. You don't take years anymore. And that's the take home message. So essentially here, this is considered as mitotic cells. So in this lesion, you have one mitotic cells, whereas this one you have four mitotic cells. Certainly, the lesion that have four mitotic cells will have faster growth rate than the one, right? So we call this is one dividing cell out of five, so 20% growth factor. This is 4 out of 5 or 80% growth factor. So growth factor, growth fraction. So usually this is low growth fraction, and this is high growth fraction. Okay, fraction is the fractions of the cell that is mitotic. So usually this one have faster growth rate than this. It makes sense. But this is observable. You can see this. All right, differentiated versus undifferentiated. So what you can see here, this is the normal hepatocyte within the liver, right? They forms these kind of like leaflet where here's a sinusoid, right? If differentiated, kind of large cells, you can see it. So in other words, they're differentiated. Right? On the other hand, if you look at the lesion called hepatocellular carcinoma, carcinoma, I mean now that is very aggressive, metastatic site issue, potentially going to the metastatic site. And you look at these cells, it's kind of hard to define what they are, right? So they're undifferentiated. What we do know is that there are some cells under mitosis, so then that they have a lot of mitotic cell, that means they're fast growing, okay? And this is really hard to treat. One of the issue with when you see lesions in the liver, it's hard to determine whether, or you always keep in your mind whether, is this the tumor that starts in the liver, or is this a metastatic site that the tumor starts from somewhere else and it comes and stays into the liver? That's something you always have to keep in mind, is that when you see a lesion, you got to make sure that it is, liver or not metastatic cell that goes to the liver. So keep in mind that differentiated cells is easier to treat than undifferentiated cells. What are some of the ways in which how the cells might move away from the initial site? Okay, there's three. You can see here's by seeding. So this is the ovarian cancer that have just kind of like, crawling to the nearby organ. We call that seeding. Or it can basically move through the distant site, through the lymphatic or through the blood. If it's more carcinoma, I would say that it is through the lymphatic. Okay. If it's sarcoma, that's like osteosarcoma, then they travel via the blood. Okay, so keep that in mind. So sarcoma or mesenchymal origin, which is mesodermal, as opposed to carcinoma, which is more epithelial. Like breast, for example, is epithelial, and they spread via lymphatic. They do some to the blood, but mainly is through the lymphatic. Grace, do you have a question?
Yeah, so they say a lot of times when women get breast cancer that they have a higher risk of getting ovarian cancer. So
Yeah.
Is this because of like seeding or any of these?
No. So I don't know if I talked to you about Mary Claire King, who has the same mentor as I do, Mary Claire King, and she was working on family with breast cancer. Right. So in other words, what she did is she looked at family who either aunt has breast cancer, the grandmother has it, that potentially the daughter might have it, and so forth. But through many generation, if you get the DNA, you can map it and say, oh, where did these people have a mutation? Obviously, it's hard work because there's a lot of changes in the DNA, and how do you know what is mutated and what is not, right? And so it's hard work to trying to sort through what does these woman who has cancer that the normal people do not have. What is it? How is it related to DNA? So in fact, it was discovered that actually she identified them. They call them BRCA, BRCA1, and now we know there's another one called BRCA2. And if you have a mutation in these two, then you have um, your increased risk of developing breast cancer as well as ovarian cancer. And so, no, we're talking about the tumor in the breast and the tumor in the ovary that become cancerous, not that the breast cancer cell has spread it to the ovary. No, we're not talking about that. That's spreading. And I don't think that you're talking to that. But the reason I'm saying that breast and cancer often go hand in hand is because they have a mutation in these BRCA1 and BRCA2. And Angelina Jolie, for example, knows that she has a mutation in BRCA1, right? And so that means that she increased risk of developing breast cancer and ovarian cancer. So what she decided to remove her breast, mastectomy, and ovaries, so ovarectomize her ovary as well. So she can't have kids, so she adopt kids. All right, fine, adopted kids. Hello. Did they have any children with Brett? I don't know. Okay, does that answer your question?
Yes, that definitely does. Thank you.
Okay, you're welcome. So different ways that cancer cell might spread to distant organs. And again, what are the three common site, distant site? The lung, the bone, and the liver. And if the tumor is derived from epithelial, like carcinoma, it usually arrive at the metastatic site through the lymphatics. If it's more like mesenchymal origin, like sarcoma, then it via the blood. Okay. Breast cancer and colorectal cancer, these are all carcinoma, epithelial. Then the invasive is to the lymphatics. And here, just basically looking at the lymphatic vessel, kind of very similar to vein, because the wall is very thin, right? There might be some here, and you can see how cancer cells are finding in there. And cancer cell, oftentimes, remember, the nucleus is going to be darker staining, right? So we also say is heterochromatic, hetero, sorry, hyper, not hetero, hyperchromatic. That means more staining, darker staining. All right. Now, the three most common metastatic sites I talked about already, uh, lungs, liver, and bone marrow, because the lungs are small sinusoids. That's where cancer gets stuck. Cancer cell gets stuck. Liver, you can see here metastatic cells, and it's hard to determine whether is this really metastatic cells, like from the breast that goes to liver, or is it maybe it is the liver cells that become cancerous like this? So how do you know? How do you know which is which? Well, if these right here express this estrogen receptor, for example, right, and the more epithelial origin, then we think it is breast, right? But it is because um, the the liver is not by any means epithelial origin. And so you won't see any cells that expresses epichem, for example. Okay, epichem is one of those uh, self-adhesion molecule that only finds in epithelial cells. So that's how, an example, you might differentiate between metastatic cell versus the liver produces on that become cancers. All right, so why don't we take I only have a couple more slides, and then we'll take a 5-minute break, and we'll come back and finish this, and then I'm going to talk about infectious disease. If I don't finish that, I'm going to finish it next week. But at the same time, this is your tough week. I'm going to post a recording on fluid dynamic, and that is because next week we have to finish those infectious disease and fluid dynamic, because um, on the 7th, on the 24th, you have your second exam. Okay, so 2 weeks from today is your second exam on those little bit on immune disorder, but typically cancer, infectious disease, and fluid dynamic. Are there any questions? Okay, let's stop here. I need to take something to drink. 5-minute break, guys.